So, I will. First of all, I'm really glad to be here today, and I will just give you a brief description of the work we are doing concerning the rational design and development of KCNT1 blocker chemotypes. This is the research group I belongs to. I'm from the University of Salerno in Italy, and I must thank anyone of these guys, these girls, for the work they do together with me. This is what I call the special K team, made up by Nunzio Iraci, that is somewhere in this call, me and Maurizio Tagliatella. We are aimed in unlock the potential of small molecule in the treatment of neurodegenerative disorders, mainly linked to potassium channels. And we are only the representatives of a huge group of researchers that is working in this field. I will show you a recent paper that we have published, and that gave us the basis for further developments. That is a virtual screening campaign using our in-house library, a molecular library of about 1,100 molecules that were screened against a homology model of the KCNT1. And starting from the docking pose obtained with quinidine used as reference compound, we were able to select 20 different compounds that were then screened using a really nice HTS assay that was developed by Maurizio Tagliatellini's group using thallium as potassium surrogate. In this way, we were able to identify 5 different molecules, namely compound 4, 13, 16, 18, and 20. That we're able to block KCNT1 at 10 micromolar and in a larger extent if compared with quinidine at a concentration of 300 micromolar. These are the structures of the molecules we have selected. And the initial selection was made on the basis of chemical diversity. We were aimed to find different chemotypes able to in modulating KCNT1 channels. Electrophysiological measurements strictly confirmed the first data obtained from the HTS phoremetric assay. Current densities of KCNT1 are strongly inhibited by the use of our compounds. And when compared to quinidine in particular, we can find that inhibitory kinetics are really much slower using these compounds. We also performed some selectivity assays, HERG, as first target of selectivity, and we interestingly found that 3 of our compounds, CPK 16, 18, and 20, does not exert any kind of blocking of HERG pigtail currents at 10 micromolar concentration. We also knew that compound 20, since it was from our library, was initially designed as KV 7.2 agonist. And thus we also performed some selectivity assays over KV 7.2. We confirmed that compound 20 is a weak activator of KV 7.2. Worth of note, we also found that quinidine also acts as inhibitor of this channel. Then my colleague Nunzio Iraci performed some dynamic simulation, trying to have some clues about the binding site and the binding mode of our molecules. We firstly performed a dynamic simulation using quinidine in our homology model. And we found that quinidine has 2 different binding poles. The first one in the panel A, where quinidine binds between the 2 S6 helices of 2 adjacent KCNT1 monomers, deeply interacting with the F346. This is reason because mutants at these specific amino acids are responsible for a loss of activity of kinidin and many other KCNT1 inhibitors. But there's also a second possible, despite less populated conformation, in which kinidin is flipped upside down and goes deeper inside the pore helix, losing the interaction with F346. And more or less similar, these two different poses are resembled by our three compounds, 16, 18, and 20, with compound 16 weakly interacting with F346. Compound 18 almost not interacting with this key residue, while CPK20 has a really strong interaction. This is also the reason why, when tested over using the F346S mutant, CPK20 is the only one significantly losing its inhibitory effect that is faintly lost by CPK16 and almost not lost by CPK18. We have also performed some preliminary in vitro pharmacokinetic analysis concerning these compounds. They are more or less all stable in human plasma, particularly CPK18 and CPK20. But when subjected to microsomal in vitro stability, CPK18 showed to be definitely more prone to metabolism, while CPK20 also showed a really high and interesting stability to this assay. Both compounds are really tightly bound to plasma proteins. This was performed. This assay was also performed in vitro, and CPK20 showed to be quite selective for KCNT1 if compared with KCNT2, and on the other side maintains similar potency when challenged over some pathological mutants, so KCNT1 in particular G288S and A934T. What we are doing now is performing some cryo-EM experiments concerning CPK20, that is the most active and metabolically stable compounds. We are also improving chemotype diversity, performing pharmacodynamic improvements of CPK20 and pharmacokinetic improvements of CPK18. And so we are also going to test these compounds by MEA over cell line bearing GOF mutation of KCNT1. We are really interested in vivo pharmacokinetic studies, and of course, we are really interested in challenging these molecules in the more relevant biological environment. I mean, mice model of KCNT1 mediated epilepsy. And at the same time, we are improving chemotype diversity. I mean, starting from the, as you can see, the green spot on the slide, starting from the compounds we have previously identified, we have also isolated all the compounds sharing chemical similarity with the firstly identified compounds. And in this way, we have been able to identify 6 more compounds that are going to subject it to the similar experimental workflow. I really thank you for your kind attention, and thank you again to give me the opportunity of this talk.
Thank you, Carmine. Amazing work from you and your team. Would anyone like to ask a couple questions?
Yes, let me ask a question. It's Greg Kazarowski. In order for these to be therapeutically interesting, you have to be able to move the potency at least 500 to 1,000-fold, okay, to get in the low nanomolar range. And that's going to take a med chem effort, probably making close to 1,000 compounds. Do you have the capacity to actually, well, first of all, do you think that's possible? And do you have the capacity to do the med chem effort? And lastly, you have to look at the selectivity in like a Eurofins panel, right? To see if it's really a viable lead. Because if you hit 40 other things in the concentration range you're starting from, historical precedent says you're not going to be able to get the selectivity you need.
Yeah, We usually collaborate with Eurofins. We are collaborated concerning KCNQ2, really, not KCNT1 till now. But I think the Eurofin panel is one of the most important panel to address selectivity issue. Perfectly right. We, from the point of view of the medicinal chemistry efforts needed to the improvement, we have all the facilities and knowledge necessary to bring forward this research line.
You really think you can make 1,000 compounds?
Yeah.
Okay. So what's the potency that you would like to see in your lead?
You mean the potency in terms of EC50?
Yeah. So I mean, how far do you think how potent do you think you need to get to be able to say that this is a useful compound in your SAR? If you only move it 10-fold, for example, based on hydrophobicity, that's not going to go anywhere. So you need to really have an SAR that shows you that you can move it 100 to 1,000-fold. Do you think that's possible based on these types of structure?
I think from our preliminary data, it could be possible, but I think most that potency, what is really important, that at least based on